Hello, I'm Essay, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
Essay. The 2004 Nobel Prize in Chemistry recognized Avram, Hershko, Aaron Ciechanover, and Irwin Rose for their groundbreaking discoveries concerning ubiquitin-mediated protein degradation. This essay embarks on a comprehensive and deeply critical evaluation of the claims presented in their collective body of work, specifically focusing on the papers which led to the Nobel recognition and the conceptual framework they established. We will delve into the intricacies of their research, analyzing the experimental evidence, the logical consistency of their arguments, the enduring impact of their findings on our understanding of cellular regulation, Section 1 Initial Observations and the identification of ubiquitin. Hershko, Ciechanover, and Rose initiated their investigations into protein turnover during the late 1970s, focusing primarily on reticulocytes, immature red blood cells, which possess an active ATP-dependent proteolytic system, but lack conventional organelles like lysosomes. Their initial experiments revealed the presence of a heat-stable polypeptide essential for this ATP-dependent proteolysis. This polypeptide, initially designated APF1 for ATP-dependent proteolysis factor 1, was subsequently purified and identified as ubiquitin, a term suggesting its ubiquitous presence across various cell types. A central claim at this stage was the absolute requirement for ATP hydrolysis in the proteolytic process and the involvement of this previously uncharacterized polypeptide. Critically, they demonstrated that inhibiting ATP synthesis abolished proteolysis, and adding back a purified fraction containing APF1 restored activity. However, at this stage, the exact mechanism of ubiquitin's involvement remained a mystery. A potential weakness, viewed retrospectively, was the initially limited understanding of the precise enzymatic steps involved, a gap that would necessitate further investigation. The earlier emphasis was on the energetic requirement, the role of a novel heat-stable factor, paving the way for future dissection of the PAR, Section 2 ubiquitin conjugation and the multi-step enzymatic cascade. The crucial breakthrough came with the realization that ubiquitin became covalently linked to cellular proteins. This conjugation, again, required ATP and was mediated by a series of enzymes. Hershko and colleagues proposed a multi-step enzymatic cascade involving at least 3 enzymatic activities, designated E1, E2, and E3. E1, the ubiquitin activating enzyme, forms a thyristor bond with ubiquitin in an ATP-dependent manner. This activated ubiquitin is then transferred to E2, a ubiquitin conjugating enzyme. Finally, E3, a ubiquitin ligase, facilitates the transfer of ubiquitin from E2 to the target protein. This model, which proposed a hierarchical enzyme system, represented a significant advancement. The claim of a multi-step enzymatic cascade was supported by careful biochemical fractionation and reconstitution experiments. By isolating and characterizing the different enzymatic fractions, they could demonstrate that each component was necessary for ubiquitin conjugation and subsequent protein degradation. The precision required to isolate these enzymes, given their relative scarcity within the cell, speaks to the methodological rigor employed. The specificity conferred by the E3 ligases was proposed as the key determinant of which proteins were targeted for degradation, a statement whose validity has been extensively confirmed in subsequent. Section 3 Identification and characterization of E1, E2, and E3 enzymes. Having established the existence of E1, E2, and E3 enzymes, the next step was to identify and characterize these proteins. While the identification and purification of E1 proved relatively straightforward due to its abundance, isolating specific E2s and E3s proved more challenging due to their diversity and lower concentrations. Early research provided initial biochemical characterization of these enzymes. The evidence at this stage largely relied on in vitro assays, measuring ubiquitin conjugation and protein degradation in reconstituted systems. The claim that E3 ligases confer substrate specificity was largely based on indirect evidence. For instance, it was observed that different E3 fractions could promote the ubiquitination of different substrate proteins. The subsequent cloning and sequencing of various E2 and E3 enzymes solidified this claim, revealing families of proteins with distinct substrate specificities. A deeper understanding of the substrate recognition mechanisms of E3 ligases emerged later through structural studies and the identification of specific protein motifs at mediate binding to target proteins. Section 4. Polyubiquitination as a signal. For proteasomal degradation, a pivotal discovery was the finding that proteins targeted for degradation are conjugated to multiple ubiquitin molecules, forming polyubiquitin chains. This polyubiquitination was proposed as the signal that directs proteins to the proteasome, a large protein complex responsible for degrading ubiquitinated proteins. The claim that polyubiquitination, rather than monoubiquitination, serves as the degradation signal was initially based on observations that highly ubiquitinated proteins were rapidly degraded by the proteasome. This was substantiated by experiments using engineered ubiquitin molecules that could not form polyubiquitin chains. Cells expressing these mutant ubiquitin molecules exhibited impaired protein degradation. The linkage through lysine 48 of ubiquitin was later identified as particularly important for proteasomal targeting. This finding highlighted the complexity of the ubiquitin system, demonstrating that the type of ubiquitin modification, in addition to its presence, dictates its biological consequence. Section 5. The 26 is proteasome structure and function. Hershko, Ciechanover, and Rose's work laid the groundwork for understanding the 26's proteasome, the primary engine of ubiquitin-mediated protein degradation. The 26's proteasome is a large, multi-subunit complex composed of a 20's core particle and 1 or 2 19's regulatory particles. The 20's core particle contains the proteolytic active sites, responsible for cleaving the target protein into small peptides. The 19's regulatory particle recognizes and binds to polyubiquitinated proteins, unfolds them, and translocates them into the 20's core particle for degradation. The claim that the 26's proteasome is the major site of ubiquitin-dependent proteolysis was supported by genetic and biochemical evidence. Mutants, lacking components of the proteasome, accumulated ubiquitinated proteins and displayed impaired protein degradation. Furthermore, purified proteasomes could degrade ubiquitinated proteins in vitro, although their initial work did not fully elucidate the intricate structure and function of the proteasome. It provided the essential foundation for later studies that revealed the complex mechanisms involved in substrate recognition, unfolding, and peptide bond cleavage. Section 6. Regulation of cell cycle and other cellular processes via ubiquitin-mediated proteolysis. The discoveries of Hershko, Ciechanover, and Rose unveiled the crucial role of ubiquitin-mediated proteolysis in regulating a wide variety of cellular processes, including cell cycle progression, signal transduction, and DNA repair. For example, they and others showed that the controlled degradation of cell cycle regulators, such as cyclins, is essential for proper cell division. The claim that ubiquitin-mediated proteolysis is a key regulator of the cell cycle was supported by experiments demonstrating that the degradation of specific cyclins is required for cells to transition from one phase of the cell cycle to the next. Inhibiting cyclin degradation resulted in cell cycle arrest. Similarly, the degradation of transcription factors and signaling molecules was shown to regulate gene expression and cellular responses to external stimuli. These findings highlighted the importance of regulated protein degradation in maintaining cellular homeostasis and responding to changing environmental conditions. The implications of these findings extended beyond fundamental cell biology. In cancer research, deregulated ubiquitin-mediated proteolysis became recognized as a contributing factor in tumor development and progression, opening avenues for therapeutic intervention. Section 7. The end rule pathway. A specific example of ubiquitin-mediated degradation. One specific example of ubiquitin-mediated degradation highlighted in the work of Hershko and colleagues involves the end rule pathway. This pathway relates the half-life of a protein to the identity of its N-terminal amino acid residue. Proteins with destabilizing N-terminal residues are rapidly ubiquitinated and degraded, while proteins with stabilizing N-terminal residues are more stable. The claim that the N-terminal amino acid residue can act as a degradation signal was supported by experiments in which proteins were engineered to contain different N-terminal amino acids. Proteins with destabilizing N-terminal residues were rapidly degraded, while those with stabilizing N-terminal residues were more stable. This pathway provided one of the first examples of a specific signal that targets proteins for ubiquitin-mediated degradation and contributed to our understanding of the mechanisms that control protein turnover. Subsequent research identified the E3 ligase responsible for recognizing destabilizing N-terminal residues, further elucidating the molecular details of this path. Section 8. Ubiquitin and disease implications for therapeutics. The recognition of ubiquitin-mediated proteolysis as a critical cellular process has profound implications for understanding and treating human diseases. Operations in the ubiquitin system are implicated in a wide range of disorders, including cancer, neurodegenerative diseases, and immune disorders. The claim that dysregulation of ubiquitin-mediated proteolysis can contribute to disease pathology is supported by a wealth of evidence. For example, mutations in E3 ligases have been linked to specific cancers. Inactivation of tumor suppressor proteins through accelerated ubiquitin-mediated degradation can promote uncontrolled cell growth. Conversely, the stabilization of oncogenic proteins through impaired ubiquitin-mediated degradation can also contribute to tumor development. The understanding of the ubiquitin system has opened up new avenues for therapeutic intervention. Proteasome inhibitors, such as bortezomib, are now used to treat multiple myeloma, a type of cancer in which plasma cells accumulate in the bone marrow. These inhibitors block the degradation of proteins, alloproteasome, leading to the accumulation of misfolded proteins and the induction of apoptosis in cancer cells. The development of other therapeutic agents targeting specific components of the ubiquitin system holds promise for treating a variety of diseases. Section 9. The broader impact and ongoing research. The work of Hershko, Ciechanover, and Rose revolutionized our understanding of protein degradation and its role in cellular regulation. Their discoveries paved the way for countless subsequent studies that have further elucidated the complexity and importance of the ubiquitin system. The claim that ubiquitin-mediated proteolysis is a fundamental cellular process is supported by its conservation across diverse species and its involvement in a vast array of cellular functions. Ongoing research continues to explore the intricacies of the ubiquitin system, including the identification of new E3 ligases, the characterization of ubiquitin modifications beyond polyubiquitination, and the development of novel therapeutic agents targeting specific components of the pathway. The study of autophagy, another major protein degradation pathway, has also been significantly influenced by the advances in understanding ubiquitin-mediated proteolysis. The interplay between these two pathways is becoming increasingly recognized as critical for maintaining cellular health and responding to stress. Section 10. Addressing initial criticisms and alternative models. While the ubiquitin proteasome pathway is now firmly established, initial interpretations and models faced criticism. Early skepticism stemmed from the novelty of the pathway, the idea that protein degradation required ATP, seemingly running counter to the energy needs associated with synthesis, was a somewhat counterintuitive concept. Furthermore, alternative protein degradation pathways were known to exist, such as lysosomal degradation, which further challenged the initial impact of their findings. A claim often levied was that the system only played a role under specialized conditions. The subsequent ubiquity of the system's involvement and regulation in processes from cell cycle regulation and immune response laid those concerns to Section 11 examination of the experimental methods and controls employed, a key strength within the work from Hershko, Ciechanover, and Rose lies in their meticulously designed experiments featuring robust controls when dissecting complex enzymatic pathways, such as the ubiquitin system. The use of stringent controls is crucial for ensuring the validity of the results. Their investigations consistently included negative control reactions lacking ATP, ubiquitin, or specific enzyme fractions. These controls served to rule out the possibility of non-specific proteolysis or other artifacts that might confound the interpretation of the data. In addition, they employed a variety of biochemical techniques, including protein purification, enzyme assays, and gel electrophoresis, to characterize the components of the ubiquitin system. A potential criticism, viewed retrospectively, is the early reliance on in vitro assays. While these assays were essential for dissecting the pathway, they may not fully reflect the complexity of the ubiquitin system in vivo. However, subsequent studies using genetic and cell biological approaches have largely validated their findings. Section 12 Evolutionary Conservation and Ubiquitin's Ancient Origins. The remarkable evolutionary conservation of the ubiquitin system across diverse species underscores its fundamental importance in cellular life. Ubiquitin itself is nearly identical in sequence from yeast to humans, indicating that its function has been stringently conserved throughout evolution. The claim that the ubiquitin system is an ancient and essential cellular process is supported by its presence in both prokaryotes and eukaryotes. While the complexity of the ubiquitin system varies across different organisms, the core components, including ubiquitin, E1, E2, and E3 enzymes, are generally present. This evolutionary conservation suggests that the ubiquitin system arose early in the history of life and has played a critical role in maintaining cellular homeostasis ever since. Comparative genomic studies have revealed a surprising level of conservation in the substrate recognition mechanisms of E3 ligases, highlighting the importance of these enzymes in regulating specific cellular processes. Conclusion: the collective body of work by Avram Hershko, Ciechanover, and Irwin on ubiquitin-mediated protein degradation represents a monumental achievement in the field of biochemistry. Their meticulous experimental approach, characterized by rigorous controls and innovative techniques, led to the discovery of a novel and fundamentally important cellular pathway. Their claims, initially met with some skepticism, have been repeatedly validated by subsequent research and have had a profound impact on our understanding of cellular regulation and disease. From unraveling the enzymatic cascade to elucidating the role of polyubiquitination, their contributions are undeniable. The ubiquitin proteasome pathway is now recognized as a key regulator of a wide variety of cellular processes, from cell cycle control and signal transduction to DNA repair and immune responses. Furthermore, dysregulation of the ubiquitin system is implicated in a growing number of human diseases, opening up new avenues for therapeutic intervention. Their work not only illuminated a previously unexplored area of cell biology but also provided a powerful framework for understanding and treating a wide range of human diseases. Their legacy endures, with ubiquitin-mediated proteolysis remaining a central focus of research in diverse fields of biology and medicine. The elegance and clarity with which their claims were articulated and supported, combined with the far-reaching implications of their findings, solidify their place as true pioneers in modern biochemistry.
We really appreciate you tuning in to this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.